It's such an honor to be involved in this evening's amazing launch for something so important. And if you're a neuroscientist who wants to understand how the brain works and what you can do to make life better for people for whom the brain is a challenge in a certain way, there is not a better place on earth that you can work than MIT in the McGovern Institute. And the addition of this amazing new Center for Autism Research makes it, it's on our shoulders to do something really, really valuable. And thank you so much for making this opportunity. So you've heard amazing talks 3 in a row from outstanding scientists linking genes to neurons to mice and marmosets. And of course, the long-term goal is to use this fantastic neuroscience to help people. And so for those of us who work on humans, adults and children, we have the advantage that we work on the species that we want to help and care about and love in our families and society. We have the disadvantage that we're very limited in what we can do in terms of developing new technologies. We have to be very safe and cautious. At the McGovern Institute, thanks to a brain imaging center, what we can do with humans, infants through adults, is remarkable compared to what it was when I was a graduate student at MIT in the 1980s. We could do nothing to look inside the mind of the brain of a child or an adult in any serious way. And now we have downstairs, thanks to the McGovern Institute, the Martino's Imaging Center, and philanthropy and grants, an amazing array of tools that measure the structure and function of the brain, both temporally, how quickly it operates, and structurally, its organization. And I'm going to focus tonight on one method that's maybe our most powerful method, but they're all powerful and valuable. Magnetic MRI or neuroimaging, which can show us things about the gray matter structure of the brain, the white matter organization, the super information highways of the brain, as well as brain function, which parts of the brain turn on, what's the sort of mental life that's supported by the biology of the brain. And because we do a lot of work with children downstairs, we have, for example, this fake MRI scanner. It's not fake news, but it's a fake scanner. It's to get children to be comfortable with the environment, to be on their back, to hear noise, to push buttons while they're on their back. Because we know that for our science to be successful, we have to have a session that's very gentle and benign to families and children. And so this gives the children all the time they want, and children with autism often are uncomfortable in novel settings, and so we give them all the time they want to get comfortable and prepared to have a good session in the scanner across the hall. And I can tell you that we often get amazing notes from children. It's a spontaneous note. We get lots of these. So here's a child who wrote, she draws a noisy scanner. Can I do that again? And she writes to the graduate student she worked with, you've let me have more fun in 3 days than I could have in any other place. Just think of it, I'm playing a game. We describe the experiments as games. When at the same time, I'm a research guinea pig. If you're a tough grader, you get to take off 2 points. And who knows, I might help someone else my age if they have any brain problems while still earning money. This is almost the ethos of the scientist, right? But it's a small amount, a modest amount of money goes to the parent as a thank you for all research participants that we do. And what do you think beats that? Nothing. It's sort of the beautiful, spontaneous language of a child. And so I've worked for many years on looking at children and their development, and the diversity of their development. And I think that all infants are born into a neurobiological lottery, where the genes they're born with and the environment they're born into will make life either easier or harder for them in a variety of ways. And as members of society, as parents, as siblings, as clinicians or researchers, we want to sort of maximize the ways in which all these children can flourish. Whatever hand they're dealt, we want to make the world as good for them as possible. And we know that so many children face challenges. The numbers are pretty remarkable, right? So I work in a number of these fields, but in dyslexia, about 10% of children struggle to learn basic reading. The current numbers in the US for ADHD are about 10%. Autism, as you know, the rates of autism are much higher now. There's a lot of issues about how to think about that, but there's a lot of children who need help. 1 in 42 boys is the current estimate in the United States. Special education in schools, 1 out of 8 children in US schools receive special education for one reason or another. Poverty in the US, different stories in different countries. 32% of children are estimated under 18 to be in families that have such low incomes. Half of the children in public schools in the US qualify for free and reduced public lunch because of low family income. And of course, all these things mix together. I mean, children can be born into better or less supported environments and have these difficulties or challenges. There's a lot of children who need a lot of help. I'm a huge believer in neurodiversity. I mean, one of the most challenging things for scientists to study the brain has been, it turns out that people are incredibly diverse, whether they have a label or not. This has been a huge source of frustration for people working on both genetics and neuroscience. You know, if we could just have identical mice, you know, in every family, we could make huge progress. I mean, honestly, if you would have said in the 1980s when I was a graduate student here, what am I doing to, am I creating a double sound? Sorry, no, okay. Sorry. If you would have said, I'm probably too close to that. If you would have said the 1980s, the amazing Human Genome Project advances we've had and the amazing advances in neuroimaging, how little those advances have yet touched the lives of people who need help, you would have been considered to be a very cranky pessimist. Now, I'm not, because I think there's amazing things just around the corner because of the science you heard about today. But a lot of that challenge has been the shocking variety that we now know underlies human brains. And it's kind of beautiful, actually. You know, we can't be reduced to simple stories of genes and brain pathways. We're incredibly complex, all of us. Genetics, experience, what age we are, what sex we are, what culture we grow up in, personality, all of these things turn out to be huge sources of human diversity in the mind and brain. And that was recognized, you know, first and foremost in many ways in autism spectrum disorder, where we realize it's such a spectrum, such a diversity that shares social and communication difficulties and some degree of repetitive behaviors. And as you know, distinctions that people thought just a few years ago were kind of useful have been wiped away in the recent psychiatric community, not because there's not huge diversity, but the boundaries of this are very unclear. And so rather than pretend that we just know the story, we just know there's tremendous diversity. And there's one child and one adult at a time. So I'm going to talk to you a little bit about human research at MIT from some of my colleagues and just a couple thoughts about future directions. So this is work from Nancy Canwisher and Carolyn Robertson showing one way to approach the human brain in autism. And they focused on GABA, an inhibitory signaling neurotransmitter. And you heard nicely from Gloria about how important it is to have enough inhibition. The right balance of inhibition and excitation is really important. And using a method called magnetic resonance spectroscopy, or MRS, Carolyn and Nancy and her colleagues focused in on GABA. So MRS gives you information about the amount of a certain chemical that's located in a certain part of the brain. And what they discovered was this, that they saw behavioral differences in people with or without autism. Those differences in visual function correlated with their severity of their clinical difficulty, so it was tied into the clinical issues. And then when they looked at GABA in this part of the brain, they saw this difference between people with or without autism. And it was somewhat specific because when they looked at another neurotransmitter, glutamate, they didn't see that difference. So this is beautiful because it's a nonverbal, translatable paradigm that could be done with animals. There's no, you know, I happen to study dyslexia. I like books and reading, but you know, those kinds of anything to do with language, we won't be able to look at in animal models very easily. But simple visual processes are highly capable of being looked at across species. This is work from Pavan Sinha, and he's developed an idea that some people with autism have difficulty in predicting what comes next. They don't get comfortable with regularities. And so here's an example of a child who was a typical developing individual. When they hear a sound over and over again, they call it habituate. They go, I've heard that, it's not a big deal. They calm down, if you want to call that, because they've heard the stimulus so often. Here is a child with autism going exactly the opposite direction, being more and more perturbed by the repeated sounds they're hearing. And so, you know, that's a thought about some of the sensory difficulties that some people with autism report. Now, this is work that I did in collaboration with Rebecca Sachs, a colleague here, and two of our postdocs who work together, the four of us. And Rebecca has been a leader in thinking about the brain basis of the theory of mind. So many of you heard about theory of mind, but let me remind you, for those of you who don't live in that part of the psychological world. So the theory of mind is the ability to attribute mental states, such as beliefs, intentions, and desires to yourself and others, and to understand that others have beliefs, desires, and intentions that are different from one's own. Children develop theory of mind, understanding that you as an adult or a parent or a sibling have thoughts that can be different from the child's very slowly. If you've ever played hide and seek with a very young child who does not imagine what you are seeing and thinking, it's pretty easy to catch them, right? They do like this, and you go, no, you're still there. And then they go like this, and they go, no, I can still see you, right? Because they don't understand what you're thinking and what you can see very easily, right? But our ability to interact with other people depends on having an understanding of what other people are thinking, even if it's different than what we're thinking. And much of the science about how this develops in young children was built around one task with a person, an evil person named Anne. These are all pretend, and a nice person named Sally. So Sally's in one room, and she puts the ball under this blanket, and then Sally leaves the room. Anne comes in and mischievously moves the ball from under that blanket into the box and closes the box. Now Sally comes back in the room, and a 3-year-old is asked, where will she look for the ball? And the 3-year-old who knows the ball is here doesn't understand that Sally can have a different thought in her head than the physical reality the child knows, and will say, Sally will look here, even though there's no reason for her to look there at all. Is that okay? She doesn't understand, that, as we all do, that people around us don't understand what's correct, even when we do. Okay? But if you move to 5 years old, the child will correctly understand that Sally believes the ball will be here, even though it's an incorrect belief. A person can have an incorrect belief that's different from physical reality. So that's theory of mind, understanding what's in the mind of another, regardless of whether it's the same thing that you know or not. Now, it's been found many times that false beliefs, this idea of understanding that somebody else can have a wrong idea in their head, by about age 5 in neurotypical children, they understand that. Children with autism typically, a huge range, wait until about age 10 until they can succeed on such a task, a huge range. But there was a mystery that we addressed, which is that adults with ASD often say this is a challenge for them, or their family says it's hard. They have a hard time understanding what other people in the workplace or at home, what they're thinking compared to what the individual is thinking. They have a hard time reading other minds. But if you test an adult on this Sally Ann task, practically everybody with autism, past 18 aces it, okay? So we had a disconnect between a scientific approach developed for children and an adult experience that's commonly felt in everyday life. And so we adopted another task that involves also some evil and even death. So we compared, how do you forgive people when something bad happens, whether they had good intentions or not? And so here's the two critical cases. There's a person, and you go in and have coffee with her, and with this evil intention, she puts poisoned sugar into your coffee, and you perish, okay? We all agree that that's bad, right? That's a bad person doing a bad thing. But what about if the same person inadvertently puts some sugar into your coffee that's poisonous, but she didn't know that? How morally culpable is that person? How much do we blame that person? Well, we think differently because they had a certain belief, the sugar was, okay, but still there was an outcome. So in this study with adults, with or without autism, we had they judged how bad something was. So everybody agreed that if you went in, somebody served you coffee, nothing happened, no bad intentions, no bad outcomes, that was okay. Everybody agreed that if somebody on purpose tried to murder you and succeeded, that was terrible. Completely the same judgments with or without autism. But here's a place where individuals look different with or without autism. So what happens if the intention was neutral? That is, you were just putting sugar in, you didn't intend to do anything bad, but by mistake the sugar was poisoned? Then individuals who are typically developing were more forgiving than people with autism. Now that's not a right or wrong answer about that, okay? And we have aphorisms for that. Sometimes we say the road to hell is paved with good intentions. That's the message, that if you do something bad, it doesn't count. If you said, I was kind of meaning to do the right thing, right? Or we say it's a thought that counts. Right? So we know that both of these things, and there's not a right or wrong answer, there's a different perspective on this, and we found this difference. And Rebecca went back and did neuroimaging studies and found that in this area that's really important for this kind of theory of mind, the right temporal parietal junction here, that she found different patterns of activation in adults with or without autism performing exactly this task. Now, what are some future directions that we can go in? So one of them, I think, builds very much on the talks you heard before. The more we understand about the fundamental neurobiological diversity that's caught within the broad spectrum of autism spectrum disorder, the more we could go back to humans and say what looks different in the brain or behavior if you have this genetic variant or that genetic variant, or different in the brain that are developed in models in mice and marmosets. So we think that's a huge area, is to understand the diversity within autism spectrum disorders. And the second thing, and I'll tell you just a word about it, is how quickly can we move to helping people? Bob Desimont's comment at the beginning, and I think other comments as well, the brain is so complicated that we have to play the long game. You know, MIT and the Government Institute and the New Center have to play the long game because it's a challenge that has to be followed through. At the same time, I'm thinking that we might be able also to think about how to make life, 10% or 15% better for people in the near future. That can also be a goal, and these goals can be coupled. So one direction that we're involved in, we're in the middle of a study. I can't tell you the findings because we're in the middle of it, and it's what's called a double-blind study. We're not allowed to know who's in what condition to keep it as objective as possible. But here's the thought. We know that oxytocin is a hormone associated with pro-social affiliative behavior in animals and humans. It makes people nicer in many experiments, okay? They're more trusting in games, if you play economic games and things like that. It makes them nicer, and there's some complications to that, but basically study after study. It reduces their response to negative stimuli. So here's an MRI study, a functional MRI study from NIH, people on placebo, responding in their amygdala to negative facial expressions, and not so much of a response once they have the medication. And a number of studies in people with autism showing alterations in brain responses when they get oxytocin compared to a placebo. So there's a number of reasons to think. Furthermore, in autism, there's atypicality in the oxytocin system. For example, a number of studies have reported reduced plasma levels. And in meta-analyses looking over many studies, although far from complete, across multiple studies, it's supporting individuals with autism in accurate emotion recognition and useful eye gaze. Although there's a paucity of study in women, that's because not only the dominance of men in the population with autism, but on top of that, because oxytocin is a hormone, it can interact in complicated ways with naturally fluctuating hormones, and people have been shy to take on that challenge research-wise, but they don't have to be. So we're involved in a study that works like this now, trying to understand both some things of the biology, but also this goal of making life 10 to 15% better for people in the near-term future. So my colleague Ode Hennon at Mass General Hospital has developed a social behavioral therapy program. These are coaching sessions, basically, where you go in, there's a program that's set up, and very systematically, in one-on-one coaching sessions, young adults with autism who don't have many sources of support in many situations are coached how to be more comfortable interacting in the workplace and social situations and so on. Before those coaching sessions, half of the participants get a dose of nasally administered spritz in the nose, oxytocin. And the idea is that they would be more, hopefully, this is the question, they'll be more comfortable in those coaching sessions with the coach to actually talk about their feelings and social skills and so on, that it might prime the pump, so to speak, to benefit from those coaching interactions. And then we're doing MRI at MIT before they go in there because we want to understand who responds to the treatment. We think, or support, we think that, as I think do many in the field, just as there's a tremendous diversity within autism spectrum disorders, so there will be in response to any given support, at least for the near-term future. It's not as if one support is going to work for the vast majority of patients. But if a support worked for, let's say, 30% of individuals pretty well, if you did the group statistic that we normally do for evaluating something, you'd say it didn't work. Because a third of people responding to something will be concluded as a failure. But if we understood who those 1/3 of people are, and then went back and did it again and showed that we know who those 1/3 of people are and we could help them, then it'd be phenomenal, right? So we want to know, does this help? Does the oxytocin enhance the effectiveness? And can we use brain measures or genetic measures to understand who will benefit from this approach, then use that approach with those kinds of individuals, but have different approaches for the other individuals. So in conclusion, we're grateful for the Center for Autism Research to understand better in humans the neural mechanisms of autism, and hopefully to accelerate support of interventions or support that will make life better in the near term as dramatic progress occurs in the kind of research you heard about earlier this evening. Thank you very much.